Good morning. I will give 2 more minutes to let everyone who's behind join us. Hello, everyone. Thank you for joining us today for ALS Webinar Wednesday series. My name is Alejandra Herrera, and I'm the marketing coordinator for ALS North America. I will be facilitating the webinar today. If you're having technical issues with our webinar platform or you have any questions, please use the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenter today is Glenn Cole. Glenn is a food safety and sanitation professional. He takes pride on developing the best teams and implementing leading industry best practices. He has been in the food industry for over 20 years and has been working at ALS for almost 1 year. Today, Glenn will be discussing environmental monitoring of foodborne pathogens in food manufacturing facilities. Glenn, I will now turn it over to you.
Thank you, Alejandra. Good morning, everybody. Like Alejandra said, we're going to be talking about some environmental monitoring of foodborne pathogens in manufacturing facilities. So today, what we are going to cover, hang on here, is why you should test, how to achieve and maintain some pathogen control, record keeping, corrective actions, and audits. So what is the goal of a good program? The most important thing is to find pathogens and spoilage microorganisms in the environment before they affect your finished product. The last thing is to assess the effectiveness of cleaning and sanitation and your GMPs in your environment. So how do you achieve Salmonella and Listeria mono control? Well, there's a few steps here: thorough cleaning and sanitation, traffic control of your personnel and equipment via designated paths between high care and low care areas, being able to deliver freight and production supplies along with removing trash from the high care facilities, separation of raw and pasteurized products, air and dust control, water control, and a good sanitation program. So where are the best places to test? The most common industry practices are zone designations. Zones 1 through 4 are the most common. There's various names out there. But zone 1 is essentially the area in which a food actually comes in contact with the production surface. It's any equipment that has direct contact with food products. The types of locations associated with this could be your filling machines, product molds, extrusion equipment, tanks, lines, hoppers, or any other equipment that has direct contact with the food. The zone 2 areas will be any equipment that has close contact but not direct contact with food. This might be legs and tables within a 5 or 10 foot radius of where a worker might be working or the production equipment might be in place. And then your zone 3 areas include auxiliary equipment in the high care areas. These tend to be the floors, the walls, the pallet jacks, drains, and other areas in which production equipment's typically used for processing. Drains especially tend to be a hotspot of microbiological activity. Everything tends to be pushed towards the drain, washed down, and that gives the organisms time to grow, food to eat, and the water necessary for it to just explode. So now the drain has all those items to promote and sustain microbial growth. It's very important part of your sanitation process. So where do you clean? I think the most important questions to ask would be, what can your employees get to? Are all the areas available to clean? Are there any difficult areas? And is there any equipment that might be necessary for maintenance to get involved with to be able to break it down so that your sanitation team can do their job? But a thorough investigative job should be completed to assess the ability to clean and sanitize all the equipment. And then the testing site should become part of a routine plan. And then the next step, you should define how often each site needs to be tested and place it on a routine schedule, and should be completed as part of your HACCP plan. If your team tests product from zone 1, then the product should be placed on hold until lab results are returned. So this is another very sticky situation that food manufacturers run into. So how do you test? Well, there's many different ways for you to be able to test. The typical methods are via swabs, sponges on handles. You use a small cellulose sponge that's attached to the end of a long plastic handle, and the whole unit is contained in a sealed bag containing a small amount of transport buffer. So you wash your hands all the way up to the forearms, use clean disposable towels to dry your hands, and using a Sharpie, you label the sample, where it came from, the day, and the person. So before you put your gloves on, you want to open the bag and use the tabs on either side and widen the opening. Once you open this, you push the swab up out of the bag and reach in there and grab it by the handle, trying to be as aseptic as possible and not contaminate anything that is already in there. So then you take the swab and you sample an area using about a 4 by 4 inch area, and you go back and forth on using one side, and then you might go up and down using the other. From that point, you want to be careful and replace the sponge back into the bag only far enough that you can grasp the sponge in one hand from the outside bag and using the other to snap the handle off. The handle will snap off. You throw that part away. You seal the bag up. Just make sure that you don't put your hand in there or contaminate any of the sample that you're trying to test for there. Roll the bag down, wrap the zip ties that are on it, and store the sponge refrigerated temperature until you're ready to send for to the lab for analysis. So then you repeat this for each sample that's collected, and you send it to the lab for overnight delivery in a cooler that is chilled with ice packs. So inside these packets, there's different neutralizing buffers. And the purpose of those buffers is to help neutralize whatever sanitizer your sanitation program is using. The most common buffers are lethane broth, neutralizing buffer, or DE broth. And each one has an advantage and a disadvantage depending upon what kind of sanitizer that you're using. So when you're talking to the lab, you need to let them know, hey, I'm using a quaternary ammonia for my sanitizer, or I'm using a peracetic acid. Because any residual sanitizer could have bactericidal activity, which could negatively affect the samples once they're analyzed. So now we move on to record keeping. So some of the most important things that you need to keep in your records are the sample log, where are you testing, when you tested, who did the testing, the day you submitted to the lab, what those results are, and if those results aren't as expected, what your corrective action is. For instance, say you came back with a presumptive on a listeria test. What did you do? Did you shut the operation down, go back, re-clean, re-sanitize, and retest? Um, does your plan say, hey, we need to find out where this is coming from? We'll shut it down and do a vector analysis. It's all those documentations where if there ever becomes a problem, you know exactly what your employees did. If you don't have a testing plan in place, you typically want to establish a baseline. So you want to collect some preliminary data, identify some sampling locations, establish your zones, define your limits, and establish the corrective actions. So for instance, you're doing an aerobic plate count, and before sanitation, your acceptable level is CFUs less than 500. But after sanitation, you want your CFUs to be less than 10 for your target, but 100 is acceptable. Anything above 100, this is where your corrective action steps in and says, hey, you need to follow your process and re-clean, re-sanitize, and retest. So what you want to do if indicators are found, thoroughly clean and sanitize all your equipment, resample the areas, re-clean and re-sanitize. Now, if you find a pathogen such as Listeria mono, Salmonella, or E. coli, you want to limit the access to the area. Most important thing is, it's there. You may not know exactly where it is, but you don't want to be able to track it anywhere else into your facility. So you want to break down and inspect your equipment in the area, re-swab the positive area after in the surroundings to determine if contamination has been localized or if it's spread somewhere. Re-clean, re-sanitize, conduct some pre-op inspections, and re-swab. You don't really want to restart any operations until all tests are negative. So once you do all this, you got to document your corrective actions against your SOP, and you got to determine if this is a one-off occurrence or if this is a repeated occurrence. So if there's if this particular spot has repeated presumptives in the area, you want to increase your frequency of sampling. Maybe you go from weekly to a daily. And then maybe after 3 weeks of negative results, you can go back to normal sampling. But if the problem persists, you might want to look at, is it a training issue? Is employee missing something? Are they cutting corners? Do they need help? Is it a facilities issue? Is there cracks in the grout? Is water and contamination able to get up under your floor structure and allow to grow that way? So it takes a lot of thought and preventative thinking in order for you to decide what to do next. And most of that is what you decide in your SOPs on the front end. So there's really many different types of testing. The microbiological testing or the indicators. Aerobic plate count is used as an indicator of bacterial populations. It's not a measure of entire growth, but it's a generic test for organisms that grow aerobically. Yeast and mold is another test that's similar, unless you know if there's any yeast, mold, or fungi that can be growing in there. pH will allow you to see the acidity of the product, and pH meters are a common tool found and used in the food processing industry. And acidity is often used as a means to preserve foods. A pH meter is required to confirm that the correct level of acidity is achieved in the product. Water activity influences the deteriorative chemical reaction rates because water can act as a solvent or it can be a reactant itself, and it can change the mobility of things through viscosity. One or combination of these factors can lead to a faster deterioration, a shortened product shelf life, or exponential microbiological growth or pathogenic growth. One of the last things on the micro side is an ATP monitoring system. And this is typically used in the field, in the production area by the sanitation team. It's adenosine triphosphate and is present in all organic materials and is the universal unit of energy used in all living cells. So what this is small little swab. You swab it on a surface that somebody has just cleaned, and it'll come back and say, it'll provide you with a number saying zeros is there's no activity, up to an unlimited number. And what it tells you is the machine is clean or it's not, because just because it looks clean doesn't mean it actually got clean. And now that brings us over to the pathogenic types of testing. So a wide variety of foods may be tested for the presence of Listeria, but processed chilled products such as dairy products, cooked meats, smoked fish make up the bulk of the samples. Composite samples from individual food lots can be prepared by blending samples into an enrichment broth and then testing. And ALS has a 24-hour turnaround time with their Solus One technology. So for salmonella, past outbreaks have usually been associated with meat products, poultry products, raw or undercooked eggs, dough, dairy products, fruits, leafy greens, raw sprouts, fresh vegetables, nut butters, and spreads, and pet foods and treats. And that's another one that ALS has a 24-hour turnaround time on it. Some foods that have been linked to E. coli would include beef, sprouts, spinach, lettuce, ready-to-eat salads, fruit, raw milk, raw flour, and cookie dough. So essentially, all these pathogenic bacteria are ubiquitous in the environment. So it's real important that you have a total plan in place that handles everything from what to do when you find it to how to eradicate it in your plant. So that brings us to the next section here is the reporting process. You know, you have all these documents, all your employees have signed off on, yes, I did this, yes, I did this. But at the end of the day, you need a quick and easy way to identify what's going on. So ALS has actually simplified this with our ALS Solutions platform. With all the sample evaluations, you can see in green, hey, we've got a 71% passing, and we've got a 27% failure rate with a 1.5% questionable rate. And we can generate this report ad hoc. We can also identify all your samples over time. Maybe there's a certain amount in a certain period. And in addition to that, we also have a visual software that will help you target and identify specific points in your facilities and map them out. And then once those results are uploaded, it will show you green, hey, these areas are good. Red, hey, we've had some issues. And we can do this in a 2D or a 3D image of your facilities. It also allows you to provide a visual system to your auditor that walks in and says, hey, let me see your records. Well, you're able to show that without having to go through binders of paperwork. I mean, you're still going to have to keep up with those, but this will be a quick and easy way. And that auditor walks in and says, hey, okay, so what's your records on table number 1 in area number 3? That allows you to be able to go out and target the exact papers that they want to look for. So this is really a comprehensive kind of overview of an environmental monitoring program. At this point, Alejandra, I don't have much else. If we have any questions, I'd be happy to answer them.
Thank you, Glenn, for the great content and your expertise on this topic. Before we begin the Q&A, we would like to ask everyone to take our survey. The link will be in the chat section. We will give you 2 minutes. We would love to hear your feedback. We will open the form for questions now. Please write out your question using the Q&A function at the bottom of this webinar. I will give everyone a few moments to type in your questions. In the meantime, I will review some brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS Global website and our ALS YouTube channel in the webinar playlist. Also, please follow our company page on LinkedIn as we post announcements and registration links to future webinars and other resources and updates. Okay, our first question coming in is from Allison. She's asking, do you have any reference methods you regularly use for air monitoring in facilities?
Hey, Allison, that's a great question. The most common method that we use would be APC plate counts with yeast and mold. That gives you the bacterial load that might be floating around. We also have some equipment that will vacuum air into and project it onto a plate, and that's how we can test to see if there's any pathogens floating around in the air. And then the other one is we can do some filtering, like air filter. We can take pieces of that and sample that out, as well as putting some of the swabs up into your air vents. Those are the most common ways of sampling and testing for air in manufacturing environments.
Okay, Kevin is asking, do you have any recommendations for increased testing during construction or remodeling?
That's a great question too. So typically during construction and remodeling, you're gonna demo an area or something like that. You're gonna seal it off pretty good, and then once you want to do some testing before all that happens, and then once you're completely done with your demolition and remodeling, and everything's settled down, and you've had your sanitation get in there and they've cleaned the rooms and all the dirt's gone, then you want to do some testing is when there's going to be no more construction, because typically your construction workers aren't really familiar with good manufacturing practices. Even though you walk them through different scenarios and you give them third-party contractors as vendors to come in facilities, they are not as in tune with your processes and procedures as your day-to-day employees are. So you have to be a little bit more cautious with them and giving them very specific paths to walk in. And then, like I said, once everything's done with the construction and remodeling, that's the perfect time to get in there and really assess if there's anything left in the environment after that remodel.
Do we have any more questions? Glenn, do you have any other comments?
Not at this time. Thanks for participating in our webinar today. If you have any questions that you think about at a later time, here's my email and my cell phone number to reach out to me anytime something pops into your mind.
Thank you, Glenn. I would also like to thank everyone for attending our webinar today. And like Glenn said, you can contact him if you have any questions. I hope everybody has a good week. Thank you.